Hi everybody, I just want to pick up where we left off. Today's class was cut a little bit short due to all the copy room issues. So let's get out your notebooks. You're going to need your composition notebook. We're going to talk over a few additional notes about heredity. Okay, so this term pleiotropy, we've already kind of hit on this before, but I just wanted to introduce this term to you guys. So the easiest way to think about this is, for instance, albinism. So albinism is a mutation on a single gene, but as you guys know, it's going to affect a lot of the ways a person looks. Their hair is going to be very, very light blonde. Their skin is not going to have any melanin, so it's going to be very light. Their eye color, so you'll never see an albino person have like brown eyes unless they're wearing contact lenses. So it's going to affect eye color. So a lot of different phenotypic traits are affected by one single mutation, okay? So that's pleiotropy. Okay, now another type of thing, this is very similar to incomplete dominance. And so polygenic inheritance, this is like skin color. So in skin color, there's these three alleles. And so if you have the dominant form for A, B, and C, you're going to be this situation. So this person would be, their genotype would be this. So the darkest possible skin color would mean that you inherited the dominant allele from from both parents. In this case, the lightest person would have inherited all the recessive alleles. But you see, if we have half and half here, the odds of these two situations are very low. So it's possible for two medium skin tone people to have a really dark child or to have a really light child. So you've probably seen that before. Sometimes twins are born, fraternal twins that is, are born with two different skin tones and possibly different from their parents. But most of the time you're going to end up with these medium skin tones, okay? Excuse me, I just had to sneeze. Okay, so polygenic inheritance just means that a single trait is sort of being influenced by a lot of alleles collectively together. All right. Okay, now I want to get back to sex linkage. So we've already done some sex link problems, as you guys know. We've done the pedigrees, but I do want to talk about some anomalies that we see with sex chromosomes. So this is just a very simple Punnett square that shows how, why there's a 50-50 chance of having a boy versus a girl. And, it seems like this could be very simple. You're either a boy or either a girl. But if you look a little bit closer and look at different examples and scenarios, you find that there are a lot of in-betweens, okay? So it's not always very clear-cut that a person is male or female. Okay, so what determines male and femaleness? Okay, so there is this SRY gene. So this is found on the Y chromosome. And the Y chromosome isn't expressed right away during fetal development. So the X chromosome is used first. So everybody has at least one X chromosome. Not having an X chromosome and, for instance, just having a Y chromosome, that's fatal. And so everybody has an X chromosome, so that's what you're going to use most of the time and when you're developing. Now, for men, this is why they have nipples, for example, because that's coded for on the X-axis, or X chromosome, excuse me. So this is an undifferentiated baby as far as the sexual characteristics, okay? And what's going to happen if you have the SRY present, okay, so that means you inherit the Y chromosome and the SRY gene is there. You're going to start to produce a lot of hormones that are going to make male characteristics develop. So these gonads will turn into testes, okay? If there is an absence of an SRY, these will develop into ovaries, and you see that the female form develops after that. But there's a lot of in between here. For example, sometimes the SRY is present, but perhaps the hormones aren't getting made. There's a mutation on the SRY gene. Sometimes the hormones are being made, but they're not being received by the cells. So remember, for the whole system of communication between cells to work, you have to have a receptor that's the right shape, you have to have a ligand that is the right shape, and it must be produced in the right way. Okay, so hormones have to be produced, you have to have the gene for it, it's got to be able to travel, it's got to be able to bind to a functional receptor, and this cell is going to have to be able to functionally respond to that. Okay, so a lot of times you could have a Y chromosome but never actually develop male genitalia. Okay, sometimes it only develops halfway, for example, and so you get sort of this intersex where you don't have a fully developed penis, but you don't have a fully developed vagina either. So that happens. Now, there's also in the case of non-disjunction. So you remember non-disjunction means that during anaphase, two chromosomes do not separate. So in this case, mom is going to make an egg with extra chromosomes in it. Okay, and one of the eggs aren't going to have any. All right, and so you can see that if mom puts an extra chromosome, an extra X chromosome into her egg, and one comes from dad, you could have a triple X female. Or you could have a female over here with only one X chromosome that was inherited from the dad. For males, they can also inherit extra X chromosomes as well. And as you guys know, we cannot have this, this condition. You need an X chromosome. Okay, so there's quite a few aneuploidies. So remember, aneuploidy means you have the not wrong, but a different number of chromosomes. And so you can see here that there's, there's females with ovaries, males with testes, but you could be XX and actually develop male genitalia. It is possible that that SRY, the SRY gene could have jumped over to the X chromosome in the man's genes. So a female could actually, develop male genitalia that way. All right, but wouldn't be able to be fertile because the gonads are still ovaries. So there's that. We were saying there's the triple X situation. That is a female. She's going to be a little taller, more at risk for learning disabilities. Klinefelter syndrome, so that's when, you have the Y chromosome, so you're a male, but that X chromosome is going to cause enlarged breast tissue and infertility. And Turner syndrome, this female would be infertile, but there are a few symptoms like a webbed neck and a few other ones. So they're really interesting to read about if you're curious about it. Okay. All right. Now, also related to the female-male thing, this is a calico cat, and you could see the spotting pattern right here. It only shows up in females. So these calico cats are always going to be female. They can never be male. And I'll go over why. Okay. So basically what's going on is there's an orange allele and there's a black allele. Okay. And so what's going to happen anytime through fetal development for a woman, she's going to have two X chromosomes, right? Okay, she's XX. Now, we don't want to overproduce a lot of the genes on these X chromosomes. So what happens is one inactivates, okay, and becomes what's called a bar body. And so one of these is going to basically shrink down into this little unusable kind of fat little chromosome, okay, and that's the bar body. Okay, so this is going to be the X chromosome that's actually used by the female. So this happens to all the cells in fetal development. All right, and so if you can imagine, if a girl is heterozygote, then let's say this cell splits in two. Okay, so mitosis happens, right? And for this cell, the black chromosome becomes inactive. And so any cell arising from this one will basically show orange first. So the phenotype of any cell originating from this one will be orange. This one, you can see the orange one balled up into the bar body. and now this black one's going to get used. So this will turn into a black batch of hair. So anything that gets passed on, this orange is going to be deactivated. Okay, and so when mitosis happens for females, whatever is the condition for this bar body versus normal condition is going to get passed on. Okay, so you can see like in this region, these are cells where the orange was inactivated, whereas the black was inactivated. So at different points in development, this X inactivation happens. And so you get this spotting pattern here. You can see it over here as well. So in males, it's very cut and dry. You're either going to be an orange cat, like a tabby cat, or you're going to be black here, depending on which type of X chromosome you inherited. And for females, if both of them are black, then it's nothing weird. One of those will turn into the bar body, and the other one will get expressed. So if you have two orange, got bar body here, all the cat's cells are going to be orange producing. But you can see for this one, some of the cells are going to inactivate the orange, some of the cells are going to inactivate the black. Whatever cells arise from those is going to determine the spotting pattern there. Okay, so that's X inactivation there. Let me know tomorrow or next class if you guys have any questions about these things.